Hidden inside the DNA of millions of Europeans is a secret handed down across generations, a defense against a virus that wouldn't emerge for hundreds of years. It's a message left behind by the dead, a gift from ancestors who lived through one of the most brutal chapters in human history. Here's the strange part. This genetic shield wasn't built with HIV in mind. It was forged during the horror of the Black Death, a plague so lethal that it wiped entire villages off the map. The people who survived had no idea they carried something valuable. They couldn't have imagined that their descendants centuries later would face a completely different virus, one this ancient mutation would happen to block. Today, roughly 1 in 10 Europeans carries this protection. And the whole story begins with people simply trying to stay alive when survival looked impossible. How scientists connected a medieval catastrophe to a modern pandemic is one of the more remarkable tales in genetics. It shows how evolution can answer questions that diseases haven't even asked, yet how nature sometimes solves problems centuries before those problems exist in their modern form. The lesson here is that the answers to some of our biggest medical challenges aren't always waiting to be discovered in a lab. Sometimes they're already sitting in our genetic code, left there by ancestors whose fight against death shaped the biological blueprint we inherited. Let's start with the mutation itself. On chromosome 3 sits a gene called CCR5, and it does something essential for the immune system. It builds a protein, a chemokine receptor, that immune cells use to communicate and coordinate their response to threats. But that same protein has a vulnerability. HIV exploits it. The virus uses this receptor as an entry point, a lock, with HIV holding the key. That's where a mutation called CCR5 delta 32 comes in. It removes 32 base pairs from the CCR5 gene. As a result, the protein either is made correctly or isn't made at all. Without it, HIV has nowhere to enter. The virus shows up at the cell door and the door simply isn't there. Inherit the mutation from both parents and you're essentially immune to HIV repeated exposure. No infection, inherit it from just one parent, and you gain meaningful resistance, lower viral loads, slower disease progression, and years or even decades of added life expectancy. It's most common in Northern Europe and much rarer near the Mediterranean, a distribution pattern that's anything but random. When researchers first noticed this pattern, they were stumped. HIV has only been recognized for about 40 years, maybe a century or two, if you stretch the timeline. So how could a mutation, this widespread one, seemingly tailor-made to resist a modern virus, have become established in populations long before that virus existed? It looked like an impossible coincidence, and it upended assumptions about how evolution operates. The breakthrough came from flipping the question, what if the mutation wasn't selected for HIV resistance at all, but for resistance to something else entirely? What if HIV, once it appeared, simply discovered the same cellular doorway that some older, deadlier pathogen had exploited for centuries? That question sent scientists digging through history for a disease brutal enough, widespread enough, to have reshaped Europe's genetic makeup within a few hundred years, something violent enough to leave fingerprints still visible in DNA 7 centuries later. When researchers first noticed this pattern, they were stumped. HIV has only been recognized for about 40 years, maybe a century or two if you stretch the timeline. So how could a mutation, this widespread one, seemingly tailor-made to resist a modern virus, have become established in populations long before that virus existed? It looked like an impossible coincidence, and it upended assumptions about how evolution operates. The breakthrough came from flipping the question, what if the mutation wasn't selected for HIV resistance at all, but for resistance to something else entirely? What if HIV, once it appeared, simply discovered the same cellular doorway that some older, deadlier pathogen had exploited for centuries? That question sent scientists digging through history for a disease brutal enough, widespread enough to have reshaped Europe's genetic makeup within a few hundred years, something violent enough to leave fingerprints still visible in DNA 7 centuries later. The obvious candidate was the Black Death between 1346 and 1353, and in recurring waves for centuries after the bacterium, Yersinia pestis tore through Europe, killing an estimated 2540 of the population in the first outbreak alone, with death tolls reaching 90 in the hardest. Hit regions, whole towns were emptied. Families vanished in days. There are historical accounts of bodies piling up faster than they could be buried. The reasoning follows naturally. If people carrying the CCR5 mutation were more likely to survive, amid this carnage, they'd have been more likely to reproduce and pass the mutation on generation after generation, as those without the protective gene died off in greater numbers. The mutation's frequency in the surviving population would climb, but when scientists actually ran the numbers, built population genetics models to test whether plague alone could explain the modern 10 frequency, the math didn't quite add up. The plague was devastating. but it struck in waves with years or decades of relative calm in between. During those gaps, populations rebounded and genetic frequencies shifted only modestly. Plague alone was quite powerful, enough mathematically to push a rare mutation to its current prevalence in just 7 centuries. So researchers looked for an additional force, something that didn't come and go like the plague but pressed on populations constantly. That search led to smallpox. Smallpox had been present in Europe for at least 2,000 years, killing roughly 3 in 10 people it infected. Unlike plagues, episodic waves, smallpox was a constant year-round threat, and crucially, it killed children disproportionately. That detail matters enormously for evolution. When a disease kills children before they reach reproductive age, it exerts far stronger, more sustained selection pressure than a disease that mostly kills adults. Parents carrying protective mutations who survived to have children would pass those genes on children without protection, would more often die before ever having children of their own. Over hundreds of years and countless generations, that kind of relentless pressure could push a protective mutation steadily upward. Many researchers now suspect it was smallpox, working alongside periodic plague outbreaks and possibly other now-forgotten pathogens that together drove the CCR5 delta 32 mutation to the frequencies we see today. Not one disease acting alone, but multiple pathogens converging on the same genetic target across centuries. In 1996, a study seemed to offer direct confirmation. In 1665, plague struck the English village of Ayam. Rather than let the disease spread, the villagers quarantined themselves entirely. A year later, half the village was dead. Researchers later tested descendants of the survivors and found the CCR5 delta 32 mutation at notably higher frequencies than expected by chance, as if the plague itself had filtered the population by genetics. The finding electrified the field, but closer analysis complicated the story. The frequencies didn't perfectly match what pure genetic selection would predict, raising the possibility that other factors, better food, shelter, or plain luck, also played a role in who survived. The real advance came from a newer technique extracting and sequencing DNA preserved in centuries-old bones and teeth. In a study from 2021, researchers examined DNA from plague victims buried in a mass grave in Ellwangen, Germany, dating to the 16th century, screening nearly 500 immune-related genes for signs of the plague's genetic fingerprint. They found real differences in allele frequencies between plague victims and modern residents of the same region. Clear evidence that plague had shaped the immune genetics of European populations, though the picture was complicated and involved many genes, not a single simple story. For CCR5 delta 32, specifically, the ancient DNA held a surprise. The mutation was already present in populations thousands of years old in the Bronze Age, even the Neolithic at frequencies similar to today. It didn't originate with the Black Death at all. The plague, at most, appears to have amplified a mutation that already existed, rather than creating it. That raises an obvious question. If the mutation is thousands of years old, why did it stay rare for so long before suddenly becoming common? One possibility is that before recurring plague and endemic smallpox intensified selection, the mutation offered little advantage, or came with trade-offs that made it less useful in earlier environments. Evolution isn't about finding a perfect solution. It's about balancing costs and benefits under the conditions of a particular time and place. The mutation's geography offers more clues. It's far more common in Scandinavia, northern Germany, and the British Isles than in southern France, Spain, or the Mediterranean one. Explanation ties back to the plague itself, which hit northern and eastern Europe especially hard in some areas, killing up to 90% of the population, which would track with higher mutation frequencies in those same regions. Another possibility involves smallpox, which was present everywhere, but may have exerted uneven pressure, with some populations facing more frequent exposure or higher mortality. Intensifying selection for the mutation over centuries in specific regions for reasons not yet fully understood. What's certain is this CCR5 delta 32 blocks HIV has existed in European populations for thousands of years and became dramatically more common in specific regions due to selective pressure from deadly diseases. Whether the primary driver was plague smallpox or some combination of forgotten pathogens, the outcome was the same. Northern Europeans in particular inherited protection against a virus that wouldn't arrive for another 5 centuries. When HIV began devastating populations in the 1980s, some people exposed repeatedly to the virus never became infected. They were carrying a gene forged in the fires of medieval plague passed down without anyone knowing what it was for. This discovery also led directly to new medicine. Understanding CCR5's role in HIV infection allowed researchers to develop CCR5 antagonist drugs that block the same doorway, turning a natural accidental mutation into a therapeutic blueprint that has extended and saved lives, especially for patients whose HIV had become resistant to other treatments. Perhaps the deepest takeaway is what this reveals about evolution itself. It doesn't plan ahead. A mutation shaped by resistance to plague was designed with HIV in mind. It couldn't have been. But when HIV eventually arrived, it ran into a barrier built over thousands of years in response to entirely different threats. That's how evolution works. It solves the problems in front of it with whatever's available. And sometimes those same tools end up solving problems that haven't even appeared. Yet scientists are still sequencing ancient DNA from across Europe, searching for other genetic fingerprints left by historical diseases. What's becoming clear is that our immune systems aren't the product of one selective event, but the accumulated result of hundreds of pathogens, each exerting pressure across generations. Some pressures still visible, others long since obscured. Some of those inherited traits may carry costs too. Researchers are exploring possible links between the CCR5 mutation and greater vulnerability to conditions like West Nile virus or certain autoimmune issues, a reminder that evolutionary trade-offs cut both ways. The CCR5 delta 32 mutation is a small case study in something much bigger. We are not separate from the diseases that have shaped human history. We carry their marks in our biology. The struggles of nameless, forgotten ancestors are recorded in our genetic code, sometimes offering protection against threats they could never have anticipated. It's an invisible inheritance, a message written in DNA, passed from the dead to the living, from a medieval plague to a modern pandemic. It's a reminder that we're connected to history, not only through the stories we tell, but through the biology we carry. Sometimes the answers to tomorrow's hardest problems were already written into our genes, long before we knew what those problems would be. All 32 missing letters of it invisible to the eye, yet powerful enough to have shaped populations, blocked a virus, and given hope to people living with HIV today. We carry the record of our ancestors' struggles, the marks of the diseases that nearly ended them, and sometimes by pure accident, the gifts they never knew they were passing on.